Hello, hi. As we have completed the algae module, we had started the survey of fungi module. Today, we will be going through the general account and asexual reproduction in Aspergillus fungi, which belongs to Ascomycota. Before we start, as usual, let us go through the route in which we will be completing this fungi module. We were to see Zygomycota, which we have already completed by seeing the life cycle of Rhizopus and Mucor. Then we have recently started with Ascomycota. In this, we were to see these many life cycles, and we have already completed Saccharomyces. Today we will be completing Aspergillus. Then we have to see Penicillium, Neurospora, Precisa and Claviceps. So after completing Ascomycota life cycles, we will be moving to Basidiomycota. Within Basidiomycota, we have to see the life cycles of Puccinia, Agaricus and Ustilago. After completing the three phylum of true fungi, that is Zygomycota, Ascomycota and Basidiomycota, we have to see Oomycota, which is the fungi-like organism. So phylum Oomycota. Under this phylum, we have to see the life cycle of Phytophthora and we have to see the life cycle of Albugo. After that, there are some general topics like lichens, mycorrhiza and fungal toxins. So let us jump into today's agenda. We are seeing Aspergillus, which is a phylum, which is a genus under Ascomycota phylum. Okay, so Aspergillus, as you can see that it is having two types of thallus. Okay, one is a prostrate thallus and one is an erect thallus, or you can say that the thallus consists of two types of mycelial habit. One is prostrate mycelium, then an erect mycelium. You can see that in this mycelial structure, you see on the area which I am pointing the mouse. Okay, this mycelium is multicellular and each cell is multinucleate with lots of protoplasm and other cell organelles, right? So during the time of maturity, what is going to happen is that one particular cell, it can be any cell, one particular cell of the prostrate mycelium will be becoming thick walled, and from this there will be the formation of an erect outgrowth. Okay, it will be up to maximum 2.5 mm height, length, only that much it will be there, but it is displaying such an erect outgrowth. And what will happen is that as the mycelium continues to mature, there will be movement of cytoplasm and many nuclei towards the tip of this erect branch. Okay, now it will be bulging because of the accumulation of nuclei and cytoplasm, and it will be getting into a globose structure. You can see it is becoming globose and it will have a thick wall outside of it. So it will be having a thick wall, and within that there will be a lot of protoplasm which accumulated from the bottom came to the top. Then it will be having many nuclei as well. So this globose or spherical structure which is seen at the top of this erect branch called conidiophore, this globose structure is called what? Vesicle. Okay, so on the top of this vesicle there will be the formation of conidias like this. Okay, and because of the function of this particular branch to carry conidia, it is known as conidiophore. So it is arising from the modified cell known as foot cell of the mycelia, and it is having a spherical head. Okay, that is called a vesicle, within which there will be many nuclei and lots of protoplasm. So this is the general morphology of it. Now we will be going to see the remaining area. Okay, when we are seeing the conidia structure, vesicle structure, you can see that there are two intermediate structures in between this vesicle and the conidia. First, yellow colored display, that is known as medullae or primary sterigmata, and from the top of this primary sterigmata there is a tubular outgrowth that is known as secondary sterigmata or phyallid. Okay, and from this phyallid, this conidia arises in a basipetal fashion. Basipetal means what? The younger conidia will be at the bottom and the matured conidia will be at the top. Okay, how this structure is formed, that will be seen as a part of our asexual reproduction discussion. Okay, so this is the general morphology. So when we are writing an answer on the structure or any question related to Aspergillus or any other genus, we have to start from systematic position, as I always say. So it belongs to class division Mycota, and it belongs to class, I mean you can say that division fungi or Mycota, okay, anything you can mention. Subdivision you can ignore. Class Ascomycetes, subclass you can ignore. Order Aspergillus, okay, then family Aspergillaceae, genus Aspergillus. Within this genus there are many species, and many species will be having many genetic features which will be helping it to adapt to diverse environmental conditions. So this is the general life cycle, okay. This life cycle of fungi has many different patterns based on the species of fungi. That means not all fungi reproduce in the same way, okay. Only some species of Aspergillus fungi will be showing. This thing I am talking only in the context of Aspergillus, okay, not all fungi. Like I'm just talking in the context of Aspergillus. Some species of Aspergillus shows sexual reproduction, while some species of Aspergillus shows asexual reproduction. We are going to see how these things happen. So I have given the notes of all things that I am explaining in different slides. You can pause the video and take it down. So Aspergillus is a eukaryotic filamentous fungi that belongs to Ascomycota. So you have to use such proper terminologies, then only you will be getting the marks when you are writing an introduction. Aspergillus is a eukaryotic filamentous fungi that belongs to Ascomycota. Then general characteristics, it's a fast growing fungi and it will be able to colonize the substrates, okay. It is feeding on the organic matter by decomposing it, so it is having an ecosystem or ecological function as well. So that's why, since they are feeding on other organic matter, they are heterotrophic organisms. Then different species of fungi, these Aspergillus, different species have varying genetic characteristics, so they are able to thrive in diverse environment, whether in alkaline environment or acidic environment or neutral environment. Then common species of Aspergillus are Aspergillus niger, Aspergillus fumigatus, Aspergillus nidulans, etc. Now the habitat means they generally grow on damp habitats. Then when we are talking about, if there is a question about the morphology or structure of Aspergillus, you have to use such format, okay. First you talk about mycelium, you can tell that, okay, there are prostrate and erect habit, and the erect thing will be conidiophores, okay. It will be, the color will be, it's a hyaline, bright or pale colored, okay, and extensively branched and septate hyphae. So we have seen this septum, okay, cells, that's why it is called multicellular. Septate hyphae, and each septate will be having a pore connection, okay, a cytoplasmic connection through a pore, so that you have to tell. Then you can say that, okay, conidiophores, okay, this thing I have already explained, okay, certain cells in the older parts of the mycelium that will be becoming thick walled, I mean 1 or 2 cells or 3 cells becoming thick walled, and these thick walls will be T-shaped. How? Can you see this T-shape? Okay, this T-shape, this is the top of the T, this is the leg of the T, you can see T-shape, okay. From this conidiophore will be arising. So I have explained all these things. Then it will be bearing a globose or elliptical or bulbous head, and that will be called vesicle. Then now the conidiophores are actually long, unbranched and aseptate, okay. You can see that these conidia are not septate, okay. The mycelium is septate, but the conidiophore is not septate. It is a cenocytic like structure which is not having any septum, and from one foot cell only one particular conidiophore is arising. It is not like a tuft form, okay. It is singly arising from a foot cell. That thing also you can mention. Now you can say that from the surface of the multinucleate vesicle, okay, from the globose structure there arise radially arranged tubular outgrowths called phyallid or sterigmata, okay. From the surface of the globous vesicle there is radially arranged, okay, throughout it without any gap, radially arranged structures called sterigmata, okay. In some species, this yellow structure will not be there. There will be only this tubular blue colored structures, and that will be bearing this particular conidia. If the yellow structure or primary sterigmata is absent, then such species are called unispherate species, okay, because it is having only one layer of sterigmata. But if it is having another layer called secondary sterigmata or phyallid, that also is there, then such species are called bispiriate species. Okay, biseriate species, because it is having 2 rows of stegmata. Okay, and from this stegmata arises the particular conidia. How it is arising, we will see as a part of the asexual reproduction discussion. So this is the notes and general morphology of this particular species, this particular Aspergillus genus. Okay, then yeah, this uniseriate and biseriate I have already explained, and as I already told, the primary stegmata is also called metullae, METULLAE. Okay, now reproductive modes, there are 3 modes. Vegetative, that is normal fragmentation, just one piece is cut and the new mycelium will be developing, that is called fragmentation. Then asexual and sexual reproduction, of which asexual reproduction we will be seeing in this lecture, sexual reproduction we will be seeing in the another lecture. So you can see that simple, it's a very simple thing. From the conidia 4, conidia will be shed to the substratum, and that will be germinating into a new particular new mycelium. Okay, sexual thing we will be seeing in the next lecture. So asexual reproduction in Aspergillus, you can see that from the foot cell there is a conidia 4. From the conidia 4 at the tip there is a globus vesicle. Then there are biseriate stegmata, primary stegmata, which are radially arranged. Okay, then without any gap. Okay, then there are secondary stegmata on the tip of which there will be formation of conidia. We are going to see how this happens, and when the conidia is shed, then it will be forming a new mycelium, and the new mycelium will be forming new conidia 4, and it will be bearing conidia, and the cycle repeats. So let us see how this conidia is formed. Okay, you can see that when the stegmata, this is a stegmata, this stegmata is actually initially oval in shape, but at the time of maturity its tip will be tapering and its tip will be pushing out. Okay, you can see that the mature phyallid or the primary stegmata thus has a narrow tapering tubular apex. Conidia will be formed within this particular apex. Okay, then you can see that initially this particular phyallid was having only one nuclei. It mitotically divided into 2. One nuclei went inside the the bulging out apex, and you can see that slowly it bulges out, like by accumulating more protoplasm. This particular apex bulges and it will be getting into its protoplasm will be taking a round form, and there will be the formation of a septum. Okay, so first conidia is formed. Similarly, second conidia and third conidia will be forming by the pushing out of the apex of this particular phyallid, and in between each conidia there will be a connection, there will be a pore connection like this. Okay, to get the nutrients from the mother or the phyallid. Okay, this is the thing. And finally, during the time of maturity, these particular segments, okay, these particular segments will be breaking off, and each conidia will be released outside. So this is the knot. So entire body, the phyallid tip pushes out that maturity. Then conidia will be formed inside the tip. Then phyallids are uninucleate. Then by mitosis, nuclei divides and enter into conidia. Then first conidium is delimited by a basal septum. This is the basal septum. Then protoplast secretes a wall, okay, a wall formation, okay, covering the protoplasm. Then it will be growing and maturing into a proper conidia. So this is the asexual reproduction diagram that you have to draw in an examination. Okay, this is the conidia 4 and vesicle. From this, one particular conidia will be shedding out. Okay, the conidia will be uninucleate in some species and multinucleate in some other species. So in the case of conidia, it will be initially will be a uninucleate structure. Then it will be undergoing multiple nuclear divisions. Okay, multiple mitotic divisions it will be having. Then it will be it's a multinucleate structure. Then when the favorable circumstances arise, when it gets sufficient nutrients, etc., it will be germinating through a proper germ tube. This germ tube will be resembling the young mycelia, which is having multinucleate, multicelled apparatus. Okay, because the conidia itself was a multinucleate structure. So this structure got multicellular, multinucleate hypha that will be germinating, that will be maturing into the vigorously branching multicellular mycelium. Okay, then this mycelium, one particular cell will be becoming thick walled and T-shaped, and from that is called a foot cell, from which new conidia 4 will be arising, and there will be vesicle and other structures. So here we are completing with this thing. Pause the video and take the notes. Thank you.